Welcome to This Explainer. Look, if you've been putting in the grueling hours studying Cambridge A-level biology, but you still feel like your marks just aren't reflecting what's actually in your head, well, you are in the exact right place today. We're doing an intense, highly visual breakdown of the exam strategies you need. We're going to deconstruct exactly what separates a pretty good answer from a guaranteed A-star answer, and we're using the examiner's very own blueprint to do it. So let's dive right in. Check this out. 9 out of 10 students get examination questions wrong in exactly this way, and it quietly bleeds away their final grade. I want to show you this mistake right now, before you make it yourself. Look at the confident student falling headfirst into what we call the generic answer trap. They write something like, "The heart revolves and pumps blood." I mean, it sounds like they generally know what a heart does, right? But to a Cambridge examiner, that's vague. It lacks proper terminology, and it is completely disconnected from any specific data. Now contrast that with the top band answer. The mammalian heart is a double pump with 4 chambers. You see the difference there? It's contextual, it's incredibly precise, and it's structurally accurate. Once you see exactly where that error creeps in, using the correct method becomes almost impossible to forget. Section 1, the high mark framework. This is our 4-step exam blueprint. We need a reliable system to consistently write those top band answers, and I'm going to reveal the exact framework examiners secretly use to award top marks. It's really not about memorizing the entire textbook cover to cover. It's about structuring your knowledge the way the mark scheme demands. Step 1 is concept identification. You have to sniff out the core biological principle hidden beneath the question's fluff. Step 2 is evidence and data extraction. You absolutely must pull specific figures directly from the provided stimulus. Step 3 is scientific reasoning. This is where the magic happens. You connect your extracted data right back to the concept using logical cause and effect links. And finally, step 4, examiner wording. You refine your entire answer using the precise command words and syllabus-specific terminology they're looking for.
Let's trace this process in action. Right here, in the Examiner Alert section, you can see it covered in red ink, basically every student's worst nightmare. Why did they lose those marks? Usually, it's context blindness. They failed to link the broad principles to the very specific scenario in front of them. Or it's technical negligence, like forgetting units or using incorrect terminology. Now follow the gear icons through the center. Concept, evidence, reasoning, and wording. This is your path to procedural mastery. If you follow these gears, you bypass that red ink entirely and generate the perfect top band answer you see down at the bottom.
Moving on to Section 2, precision in data extraction. Let's see how this builds into real, tangible skills. This is the critical jump from just knowing generic textbook definitions to extracting hard mathematical evidence. Top-performing students don't just know the facts. They demonstrate a connected understanding by grounding their concepts in real data. So you must avoid superficial graph interpretation. Please don't just write, "the line goes up." You have to extract the precise data points from the tables or graphs to prove it. And you know what the most heartbreaking mistake of all is? The absolute panic of forgetting your units. Always, always check and include your units. Let's zoom right into a classic biological graph to see this, Vmax and the Michaelis-Menten constant, Km. Notice the flawed student answer highlighted in red. The enzyme speeds up the reaction until it reaches a maximum speed. Km is where the enzyme works at half speed. Literally just a generic definition, zero data application. Now look at the top band answer in green. As substrate concentration increases, reaching Vmax at X micromoles per minute, the Km calculated at half Vmax is Y micromoles, indicating high affinity. It is totally backed by data, it uses rock-solid scientific reasoning, and it includes the specific units extracted right from the axes of the graph. That right there, that is your A-star blueprint. Applying this exact same logic to anatomy, let's take a look at the mammalian heart. The examiner red flags on the left are glaring. The flawed answer says, "My heart revolves," and uses completely inaccurate labels, totally confusing cause and effect. It's just a textbook definition dumped onto the page with zero context. But look at the improved answer in the center. It connects the understanding. It actively separates oxygenated and deoxygenated blood, and it explains that this structure ensures efficient pressure maintenance. It applies fundamental biological principles directly to the anatomical evidence. All right, Section 3, mastering the method mark. This is your ultimate safety net for when things get tricky.
Look, even if your final calculation goes completely off the rails, examiners actually award marks for the process itself. But to capture these method marks, you need a procedural sequence. 1. Deconstruct the question. 2. Extract the data. 3. Select your analytical framework, like making sure you choose the right statistical test. 4. Execute with extremely clear notation. If you write your steps out clearly, you catch those partial marks even if you stumble at the finish line. Let's actually see this safety net in action with statistical analysis. In this spot the error section, the student did a calculation, wrote down 3.7 times 12, then 3.7 times 5, and ended up with a completely wrong final calculation of 0.35 times 16. Ouch. But look at the improved answer next to it. They laid out the correct mathematical steps beautifully. 0.175 plus 0.68, clearly showing the flow to 0.855. Even if that final decimal was a nervous typo in the exam hall, those clear method steps guarantee partial credit. The takeaway, always show you're working. Section 4, spot the examiner red flags. Let's raise the stakes a bit. We're moving into a quick interactive challenge to test your ability to spot examiner traps in real time. Okay, rapid fire challenge for you. Imagine you've got a respiration experiment right in front of you. What actually happens when the substrate concentration is doubled? Take a look at your mental wet lab notebook. Can you spot the anomaly? Think about it and lock in your answer right now before I reveal the examiner's verdict. And here is the reveal. We're investigating respiration using redox indicators like DCPIP and methylene blue. Look closely at the wet lab notebook table on the left. The color of DCPIP goes from blue to decolorizing to colorless. Seems fine, right? But what are the examiner red flags on the right? It's the dreaded units trap. Principal examiner reports highlight year after year that students bleed easy marks simply by forgetting to check units in calculations or failing to accurately record data anomalies. If your notebook just says it turned clear without specific timing and concentration units, well, you've hit the generic answer trap all over again. I honestly cannot stress this enough. This is a direct quote from the Cambridge Principal Examiner Report. Answers are often too vague. Examiners want specific details. Students forget to check units and terminology. Precision isn't just nice to have, it is absolutely non-negotiable if you want the highest marks. Section 5. Securing top band answers. We've covered the pitfalls, so let's move up to the final tier of mastery, the highest stakes questions involving complex genetic and cellular mechanisms. At this level, higher-order assessment objectives demand that you do more than just describe what's happening. You have to evaluate it. You need cross-topic integration. Think connecting respiration directly to enzyme kinetics. You must identify limitations and assumptions in the experimental data, and you have to confidently evaluate alternative interpretations of that evidence. Let's apply this perfect framework to the Calvin cycle in the light-independent stage. Look at the spot the error box here. The ATP concentration goes down because it is used in the Calvin cycle. It seems logical enough, but it gets crossed out. Why? Terminology neglect. A top band answer starts with a biological concept, supports it with specific data pulled from the prompt, and concludes with scientific reasoning using proper examiner wording. You can't just vaguely gesture the Calvin cycle. You have to name the precise steps and the specific molecules involved. Finally, focus really closely on the examiner red flags on the right side of this gene regulation diagram. Students consistently lose marks by giving general answers rather than specific details. They confuse fundamental terms. Actually, scratch that, it's worse. They literally swap the word gene for allele, or write gene when they specifically mean allele. And the biggest red flag? Ignoring the context entirely. A high-scoring answer tailors the concept of structural versus regulatory genes specifically to the case study provided in the exam, not just some memorized paragraph from a textbook. All right, to lock this all in, here is your ultimate exam day checklist. Memorize the sequence. Action 1, identify. Find the core biological principle. Action 2, extract. Pull specific figures and units directly from the data. Action 3, reason. Connect that data back to the concept logically. And Action 4, refine. Use precise command words and terminology. This is the exact step-by-step action plan you need to take into the exam room. So, it all comes down to this. When you sit down and turn over that exam paper, are you going to fall into the generic answer trap? Or will you execute this 4-step framework with the mathematical and linguistic precision needed to unlock your A-star? The blueprint is literally in your hands now. Thank you so much for joining this explainer. Step into that exam hall with precision, subscribe to Ace Your Exams, and we'll see you next time.